member of Alverno's iGEM team.
The acronym iGEM stands for International Genetically Engineered Machine. iGEM started at MIT and runs the premier student competition in synthetic biology. Student teams are given kits of biological parts and work in the summer to create biological systems and run them in living cells.
Synthetic biology picks up where biotechnology leaves off. Synthetic biology is engineering genetic sequences by making genes like Lego bricks and DNA into a programming language. It is standardizing genetic sequences and using them to build useful things, just like an electrical engineer uses things like capacitors, transistors, and resistors in order to make an electrical system.
Synthetic biology has many exciting applications in medicine, biosensors, materials, biological research, and computing. For example, synthetic biology can use genetic information to formulate vaccines and other treatments. Another potential application includes efficient production of biofuel. However, it is difficult to create these complex synthetic biology devices. Making complex DNA circuits requires isolating independent genetic components. Programming cells often requires building circuits of several genes together on the same piece of DNA. Genes expressed on the same plasmid can interfere with each other's expression. Bioengineers have observed that when two genes are placed next to each other, they often unexpectedly interfere with each other's expressions. The cause of this may be supercoiling, which directly affects the expressions of genes, turning them off or on depending on the direction of the supercoil. Our research addresses the issue of gene interference in multiple gene circuits, specifically the issue of supercoiling.
Transcription can lead to supercoiling. Essentially, what supercoiling is a double helix twisting so much so that it coils onto itself. When you want to transcribe two genes on the same piece of DNA, it leads to two types of supercoiling: positive and negative. Positive supercoiling is putting so much tension on the double helix that it makes it harder to transcribe the gene. Negative supercoiling puts less tension on the double helix, making it much easier to transcribe the gene. This means that supercoiling makes transcription a lot less predictable.
The goal of our project is to assemble many different versions of these genetic circuits you just saw in the earlier picture. These genetic circuits consist of two or more genes that each perform a task. The genes can be facing in different directions, which direct their transcription in order for them to perform their task. However, because of supercoiling, they end up messing up each other, so they can't perform their tasks. So our goal as a team is to fix this problem. To do this, we propose a couple different inserts in between the two genes, including base pair spacers of different lengths, which we think will absorb the coiling, clamps, which will clamp between the two, and a terminator spacer as a final testing factor. Finally, the two genes are assembled into a genetic circuit.